Okay, thank you, Tan, for the introduction. So today I'm going to kick us off by starting off in the adolescent end of the lifespan spectrum, looking at the role of sleep in brain maturation and cognitive development. Okay, I have no conflicts of interest to disclose. So we know adolescence as a period of rapid development where many biological, cognitive, psychological, and social-emotional changes occur in preparation for adulthood. Cognitively, there is an increase in crystallized intelligence together with processing robustness and speed and fluid intelligence from childhood to adolescence before stabilizing in adulthood and then tapering off later on in life. Synaptic density in the cerebral cortex, on the other hand, does increase rapidly after birth, but instead of continuing to increase in adolescence, it actually drops sharply before stabilizing in adulthood. This is a figure by Peter Huttenlocher, who painstakingly demonstrated this by counting synapses in post-mortem human brains from a newborn to a 95-year-old. And this paradoxical decrease in synapses during adolescence is now commonly described as a pruning process, where much like pruning a rose bush, pruning of weak connections in the brain occur to strengthen strong ones and improve overall efficiency of brain networks. Now, in living brains, we can similarly observe this trajectory with structural MR scans, where cortical gray matter is observed to prune in a posterior to anterior fashion, starting in the primary sensory regions of the visual cortex and sensory motor areas, followed by the parietal, frontal, temporal heteromodal association sites, and culminating in the prefrontal regions. Now, given that this is a neuroimaging community, I think most of you are familiar with these aforementioned changes. But what happens to sleep during this period? If you record EEG from a sleeping individual, you will notice that as one transitions into a deeper state of sleep, the frequency of brain oscillations reduces, so they get slower, and the amplitude increases, so they get larger. And these slow oscillations, or slow waves, particularly in the 0.6 to 4 Hz range, dominate in deep sleep and are thought to represent sleep intensity as it increases following a period of extended wakefulness, particularly in regions that were most used during prior wake. Interestingly, the trajectory of sleep slow wave activity, or power, also mirrors developmental changes in synaptic density, increasing in childhood and then decreasing rapidly in adolescence. And this is thought to reflect the fact that in younger compared to older adolescents, higher waking activity and higher synaptic density necessitates higher downscaling processes during sleep, which is then reflected as increased slow wave activity. Notably, the topography of maximal sleep slow wave activity also shifts from posterior to anterior regions, mirroring changes in cortical gray matter development described earlier. And these changes are only localized to the slow wave activity band and don't occur in other frequency bands. Now, as prior work has sought to establish associations between age and brain structure, sleep slow wave activity, and cognition, in the present work, I sought to confirm this in mid to late adolescence, as well as examine whether cortical thinning as well as reductions in sleep slow wave activity together mediate these age-related changes in cognition observed. So to do this, we took 114 adolescents aged 15 to 19 years from a series of sleep studies that our lab conducted over a 5-year period, and you can find out more details about these studies at this website below. Cognition was assessed in domains of nonverbal intelligence, speed of processing, working memory, and sustained attention. Sleep EEG was recorded over a 9-hour baseline night, and we had slow wave activity power average across this night. Finally, structural MR scans were taken 2 weeks before to 2 months after the baseline sleep EEG session, and then cortical thickness measures were computed with FreeSurfer. So onto results, we found that cognitive functioning did increase with age, but only for domains of nonverbal intelligence and speed of processing, as likely domains of working memory and sustained attention had already reached maturation by this age. In addition, we replicated prior findings showing reductions in cortical thickness and slow wave activity power even in this very narrow age window. To conduct mediation analysis, we first sought to identify a region in the brain that was simultaneously associated with age, slow wave activity, and cognition, and this region was identified to be in the bilateral middle temporal regions. And in serial mediation models, we showed that the age-related improvements in cognition were mediated by bilateral middle temporal regions together with reductions in slow wave activity. So far I've talked about how sleep does not only mirror brain maturation levels, but also together with brain structure and mediate age-related changes in cognition. And this appeared to occur in the bilateral middle temporal regions, which could be more sensitive to developmental changes during this 15 to 19-year-old period. So in the rest of my talk today, I wanted to discuss whether sleep could just be a mirror or mediator of brain maturation, but also a potential modulator. And to do this, I took data from the ABCD cohort, where there was sleep data, brain structure data, and cognition data in a group of 9 to 11-year-old children, and 2 years later. And I did this from a 24-hour movement behaviors perspective, because it's not just what you do at night that matters, but what you do the entire day. So what are these 24-hour movement behaviors? Sleep, sedentary behavior, and physical activity are movement behaviors that are thought to lie on a continuum, with sleep on the one end with very little or no activity, and vigorous physical activity on the other end. And at any point in a 24-hour day, we would be engaged in an activity type along this continuum. While traditional guidelines only focus on promoting each behavior in isolation, for example, promoting moderate to vigorous physical activity, this only accounts for 1 hour of a typical 24-hour day. What about the remaining 23 hours? These activities are likely to be codependent both within and between days. In 2016, a Canadian task force established the first 24-hour movement guidelines for children and youth aged 5 to 17 years, recommending that they get at least 60 minutes of moderate to vigorous physical activity a day, 9 to 11 hours of sleep, and no more than 2 hours of sedentary behavior, which includes screen time. And this infographic on the right here, on the left here, is quite clever because the height of these bars is supposed to represent how much time proportionally we should spend on each activity in a 24-hour day. So as mentioned, we took data from the ABCD study where there were 11,878 children aged 9 to 11 years at baseline. They had brain scan data, cognitive tasks, and questionnaires taken at least every 2 years. And the present analysis was taken from Release 4.0, where we had baseline and 2-year follow-up data. So going back to our question, we wanted to ask if adherence to these 24-hour movement recommendations, so moderate to vigorous physical activity of at least 60 minutes per day, screen time no more than 2 hours a day, and sleep of 9 to 11 hours a night, related to cognition and brain structure at baseline and 2 years later. Cognition was assessed with the NIH toolbox, assessing domains of language, episodic memory, executive functioning, attention, working memory, and processing speed, while brain morphometric measures were taken from free surfer, total cortical, and gray matter volumes in the 4 major lobes. Physical activity assessments were taken from the Youth Risk Behavior Survey, asking during the past 7 days, how many days were you physically active for a total of at least 60 minutes per day? And only children who answered 7 days were deemed to have met the activity recommendation. Sleep assessments were taken from the caregiver-reported sleep disturbance scale for children, asking how many hours of sleep does your child get on most nights? And only children who slept 9 to 11 hours a night were deemed to have met the sleep recommendations. Finally, for sedentary behavior, this was taken from the Youth Screen Time Survey, asking on a typical weekday or weekend how many hours are spent on recreational screen time activity. And only children who answered 2 hours or less were deemed to have met the screen time recommendation. And I think that's a bit cut off here. Okay, so looking at the percentage of children meeting the 24-hour movement guidelines, at baseline, we saw that only 4% of children were meeting all movement guidelines, while 31% did not meet any. 2 years later, the percentage of children meeting all guidelines was only 3%, while a staggering 43% now did not meet any of the movement guidelines. So in baseline cross-sectional analysis, we saw that compared to children who did not meet any movement guidelines, children who met screen time guidelines, sleep and screen time guidelines, and all guidelines had in fact better fluid and crystallized intelligence scores, as well as on a composite cognition score that took into account the average of 5 tasks common to both baseline and follow-up time points. For cortical gray matter volumes, Only children who met sleep and screen time guidelines had greater cortical gray matter volumes, particularly in the frontal and temporal lobes, than children who did not meet any recommendations. So we repeated these analyses 2 years later and found that across all associations examined, only children who met sleep and screen time guidelines had consistently better cognition and greater total gray matter volumes than children who met no guidelines. There were also some negative associations here with activity that I will discuss at the end of my talk. So just focusing on these two guidelines, sleep and screen time, we saw that 5% of children met both guidelines at both time points, while 77% did not meet either at either time point. And again, when comparing these two groups, we saw that children who met sleep and screen time guidelines at both time points had greater improvements in cognition, as well as greater gray matter volumes 2 years later. And because they were already on the pruning end of the spectrum, this would imply less pruning in these individuals, and perhaps an optimal level of pruning is required for healthy neurodevelopment. So in the second part of my talk, I discussed how children who met sleep and screen time recommendations had better cognition and greater gray matter volumes than those who met none, and these relationships persisted both cross-sectionally and longitudinally 2 years later. So some discussion points. We wondered if increased screen time could have negated effects of just meeting adequate sleep recommendations alone. We know that screen time, particularly too close to bedtime, could increase emotional and physiologic arousal, and that could lead to poor quality sleep and then poorer cognition. In addition, excessive screen time could have also reduced time available for educational activities. We already know that children who reported spending more time on their screens also reported less time sleeping, and so this could also have reduced time available for other activities during the day. Alternatively, these results could also reflect greater parental awareness or ability to supervise in children with controlled screen time. And at least one study has shown that positive parenting can reduce the impact of socioeconomic disadvantage on the development of brain structure in adolescents. Finally, we also saw that there were null or even negative effects of meeting activity recommendations, and a closer look at the data showed that children who tended to report meeting activity recommendations all days of the week tended to also have greater externalizing behaviors, so perhaps physical activity was used as a coping mechanism to deal with some of these behavioral disorders. Okay, so in summary, today I've talked about how EEG sleep-slow activity mirrors developmental cortical restructuring and functioning, and in mid to late adolescence, this restructuring appears to be strongest in the middle temporal regions. Reductions in cortical thickness together with slow wave activity together mediate the increase in cognition, and this could represent increased network efficiency, particularly for logical reasoning and speed of processing at this age. And then finally, sleep together with controlled screen time appear to also modulate cognitive and cortical development in the transition from childhood to adolescence. Okay, I'd finally like to thank everyone involved in this study, especially our lab PI, Professor Michael Chi, our funding and data sources from the ABCD study. And if you'd like to know more details about this study, you can also scan these QR codes here. Okay, thank you for your attention.
Given that we have 2 more minutes, maybe we can take one question, and then we'll keep the rest of the questions at the end. Okay, there's no question from the poll, but I had a question of whether the fact that the activity is not, meeting the activity requirements is not associated with a positive outcome. Is this because those people who had more activity also tended to have less time for sleep? Did the activity was mostly conducted at night?
Yeah, I didn't look at that. I looked at sleep and screen time, but not activity. But it's possible. Also, children who were meeting activity recommendations on less days of the week, so I think like 2, 3, 4, 5, and 6, they who actually did have better cognition. So it was only the group with 7 days of activity that tended to have worse cognition and also greater externalizing problems. So okay. Maybe those would be related somehow.
Okay, yeah, maybe they drove the negative effects.
Yeah, and also I think that there's been like a recent revision to the guidelines to say that, if you have at least 60 minutes in a day, that could still be beneficial. It doesn't have to be every day of the week. So maybe, that could be in support of those guidelines.
Thank you. And maybe one question from Renata?
I'm yelling.
Yes,
no,
it
thank
works.
you. In adolescence, another thing that is changing is the circadian rhythm. Did you, by any chance, have any data on this? Because adolescents become late-night persons, which adds to the fact that they get a lack of sleep, for example. Did you have any circadian data on these children?
No, I don't know if the ABCD has chronotype data. Does anyone know? No, I
just
didn't
might
look
be
at that,
interesting to cross-link your findings with the changes in the circadian patterns in these children.
Yeah, definitely. Thank you.
Thank you.